Hello friends, welcome to chapter number 38 SDS page electrophoresis. SDS polyacrylamide gel electrophoresis. चलिए देखते हैं क्या है इस चैप्टर में न्यू, कुछ इंटरेस्टिंग समझने का प्रयास करेंगे. When proteins are separated by electrophoresis through a gel matrix, a smaller proteins migrate faster due to less resistance from the gel matrix. Other influences on the rate of migration through the gel matrix include the structure and charge of the proteins. Polyacrylamide gel electrophoresis involves separation of protein on the basis of charge and molecular size. Acrylamide is soluble in water, and upon addition of water, it polymerizes, resulting in formation of polyacrylamide. By using this method, the pore size in the gel can be regulated. Increased concentration of acrylamide results in decreased pore size after polymerization. Polyacrylamide gel with a small pores allows the smaller molecule to enter the pores and travel through the gel, while large molecule get trapped at the pore opening. Figure 97. Direction of protein migration: cathode, buffer, protein band, एनोड. और ये है वेल. लार्जर हाई मोलिकुलर वेट प्रोटीन, स्मॉलर लो मोलिकुलर वेट प्रोटीन, और ये है जेल. As with all forms of gel electrophoresis, molecules may be run in their native state, preserving the molecule's higher order structure. This method is called native PAGE, polyacrylamide gel electrophoresis. Alternatively, a chemical detergent may be added to modify the structure and thus turn the molecule into an unsaturated molecule whose mobility now only depends on its length. Molecular weight. One of the common denaturation agents used is sodium dodecyl sulfate, SDS, sodium dodecyl sulfate, SDS, and the method is known as SDS page electrophoresis. SDS is a detergent with a strong protein denaturing effect and binds to the protein backbone at a constant molar ratio. In the presence of SDS and a reducing agent that cleaves disulfide bonds critical for proper folding, proteins unfold into linear chains with negative charge proportional to the polypeptide chain length. At the pH at which gel electrophoresis is carried out, the SDS molecules are negatively charged and bind to proteins in a set ratio. Approximately 1 molecule of SDS for every 2 amino acids. In this way, the detergent provides all proteins with a uniform charge to mass ratio. By binding to the proteins, the detergent destroys their secondary, tertiary, and or quaternary structure, denaturing them and turning them into negatively charged linear polypeptide chains. Effects of SDS on the formation and charge of a protein. When subjected to an electric field in PAGE, the negatively charged polypeptide chains travels towards the anode with different mobility. Here you can see the completed and stained SDS page electrophoretic showing separation of a test protein sample depending on their molecular weight. On the left is the control run of proteins with different molecular size for determining the molecular weight of unknown sample. Picture of an SDS page. The molecular marker ladder are in the left lane. ओके फ्रेंड्स, ये चैप्टर कंप्लीट हो चुका है। नेक्स्ट चैप्टर है चैप्टर 39 पॉलीमरेज चेन रिएक्शन। पॉलीमरेज चेन रिएक्शन में क्या करते हैं इसके बारे में समझते हैं। पॉलीमरेज चेन रिएक्शन और PCR was developed in 1983 by Carrie B. Mullis, an American biochemist who went on to win the Nobel Prize for Chemistry in 1993 for his invention. Before the development of PCR, the methods used to amplify or generate copies of recombinant DNA fragments were time-consuming and labor-intensive. In contrast, a machine designed to carry out PCR reactions can complete many rounds of replication, producing billions of copies of your DNA fragments in only a few hours. PCR is an in vitro method that allows a single short region of your DNA molecule, even a single gene, to be copied multiple times by tag polymerase. It is based on the natural process a cell uses to replicate a new DNA strand. Only a few biological ingredients are needed for PCR. The integral component is the template DNA, that is the DNA that contains the region to be copied, such as a gene. A little as one DNA molecule that can serves as a template. The only information needed for this fragment to be replicated is the sequence of two short regions of nucleotides, the subunits of DNA, at either end of the region of interest. These two short template sequences must be known so that two primer short stretches of nucleotides that correspond to the template sequence can be synthesized. The primer bind or anneal to the template at their complementary site and serves as the starting point for copy. DNA synthesis at one primer is directed towards the other, resulting in replication of the desired intervening sequence. Also needed are free nucleotides used to build the new DNA strand, and a DNA polymerase, an enzyme that does the building by sequentially adding on free nucleotide according to the instruction of the template. It is a three-step process that is carried out in a repeated cycle. The initial step is the denaturation or separation of the two strands of the DNA molecule. This is accomplished by heating the starting material to temperature of about 94 degrees Celsius. Each strand is a template on which a new strand is built. In the second step, the temperature is reduced to about 55 degrees Celsius so that the primer can anneal to the template. In the third step, the temperature is raised to about 72 degrees Celsius and the DNA polymerase begins adding nucleotides onto the ends of the annealed primer. At the end of the cycle, which lasts about 5 minutes, the temperature is raised and the process begins again. The number of copies doubles after each cycle. Usually 25 to 30 cycles produced a sufficient amount of DNA. Step 1. Denaturing 95 degrees Celsius. Parent DNA, DNA template, strand, DNA primer to DNA strand. Step 2. Annealing 55 degrees Celsius. Section of DNA to be amplified. Nucleotide DTTP, DCTP, DATP, DGTP. Step 3. Synthesizing 72 degrees Celsius. टैग पॉलीमरेज और ये है रिपीट साइकिल 20 to 40 टाइम्स न्यू डीएनए स्ट्रैंड रेडी हो गया। फिगर 100 बेसिक पीसीआर प्रोग्राम। Initial denaturation for 2 minutes at 94 degrees Celsius. This initiation step heats the double strand DNA template strain to the point where the strain starts denaturing and the hydrogen bonds are broken between the nucleotide base pair. The initial denaturation steps is commonly performed at 94 to 98 degrees Celsius. Second, denature 30 seconds at 94 degrees Celsius. Continued denaturation of double-stranded DNA. Third, anneal primers for 30 seconds at 55 degrees Celsius. The forward and reverse primers are stable within this temperature range to anneal to each of the single standard DNA template strain. The DNA polymerase is also stable enough to now bind to the primer DNA sequence. Fourth step, extend DNA for 1 minute at 72 degrees Celsius. The tech polymerase has an optimum temperature around 70 to 75 degrees Celsius, so this step enables the DNA polymerase to synthesize and elongate the new target DNA strand accurately and rapidly. Step 5, repeat step 2 to 4, 25 to 30 times. Final 6 step, final extension for 5 minutes at 72 degrees Celsius. A final extension to fill in any protruding ends of the newly synthesized strands. Because DNA from a wide range of sources can be amplified, the technique has been applied to many fields. PCR is used to diagnose genetic disease and to detect low level of viral infection. In forensic medicine, it is used to analyze minute trace of blood and other tissues in order to identify the donor by his genetic fingerprint. ओके फ्रेंड्स, नेक्स्ट चैप्टर है चैप्टर नंबर 40 डीएनए आइसोलेशन. इसे हम लोग नेक्स्ट वीडियो में कवर करेंगे. तब तक के लिए पढ़ते रहिए, मुस्कुराते रहिए. बाय बाय, मिलते हैं नेक्स्ट वीडियो में. थैंक यू.